Och nu då, Efthalia. Hello. Congratulations.
Thank you so much.
Thank you.
My Swedish are not so good as well, so I will speak in English.
Yeah, you try this one as well then.
Yes, I do. Yes. So hello everyone, I am Efthalia. I'm a part of the Blomgren group and I would like to give you a brief summary of my project of how we can leverage artificial intelligence to identify biomarkers for microglia for neurodevelopmental disorders and brain injuries. So microglia are, I think most of you know, the first line of defense in our brain, the immune cells of our brain. They are cells of embryonic origin and they go to our brain and stay there throughout our whole lives. They have several functions. They maintain the brain homeostasis. They survey the brain parenchyma. They're like the police of the brain. They eat dead cells and debris, and they clear out synapses that are not necessary anymore. So in real life, microglia look like that under homeostatic conditions. So they have very nice, long processes, ramified. But in case of injury, disease, or illness, then they react. They adopt a reactive phenotype, which can be either neuroprotective or neurotoxic. And their morphology changes as well to an either amoeboid or to a round morphology, where they start eating. They take up cells in them and they kaput them. So microglia are so important because they're actually involved in many, many different neurological disorders in children and young people, from brain cancer to schizophrenia, epilepsy, and also cranial radiotherapy, which is an example that I will give you today some results about. So we decided to look at the microglia long-term in the mouse, in the healthy and injured hippocampus. So the hippocampus, as many of you know, is an area deep in the brain, and it's a key brain area for memory and learning. And we use a technique called single-cell RNA sequencing, which is a large-scale technique. And for everyone who is not familiar with it, it's basically we can dissect all the cell types that are in the brain tissue. So like a fruit salad, we can look at the strawberries, the kiwi, the pineapple, everything by itself and find the changes. Yes? Oh,
it's upside down.
Sorry. Technical issues. Thank you. So the fruit salad, in the case of microglia in the brain, it will look like that. So these are the healthy microglia, and with different colors, you see microglia from different developmental stages. So we have microglia from 3-week-old mice, so adolescent mice, up to 1-year-old mice. So in a human, it would be approximately 50 to 60 years old of a human. And we can basically use our young microglia to predict their developmental stages to the older microglia. However, in the cases of injury, such as radiation-induced injury, we see that this nice separation here, it's actually lost, and we see that the injury plays a very, very big role into the development of our microglia. Artificial intelligence, as many of you know, how many of you are familiar with ChatGPT? Yes, exactly. So it has revolutionized the way that we look at biology, the way that we look at medicine. So basically, for our case, we wanted to use all of our single-cell RNA sequencing data and combine them with different kinds of artificial intelligence, such as machine learning or deep learning, and teach basically models to react like microglia under different conditions. So this is an example of how a model in deep learning looks like. Don't get lost in the mathematics. Basically, we teach our models for different conditions, such as healthy conditions or injury conditions, such as irradiation, and then we predict the developmental trajectories at each different time point. Our goals are to create holistic AI models using microglia from different disorders and different species. For now, we have mice, but we plan also to use human data. These models benefit from a lot of data, so they learn better the more things that they look at. We want to improve our understanding of brain development and immune responses, which will lead to acceleration of target screenings and drug development, and also identification of biomarkers. And finally, we want to have more personalized and effective treatments for young people. And as all of you here, we want to actually give children a better chance at a healthy future. And my acknowledgements, I would like to thank the Blomgren Kingdom and my supervisor Anamantio, who is here, but also the Kaylis Lab that helped us a lot with the AI part of the project. And you for giving me the opportunity to continue with my research. Thank you.